See, it's a cooperative effort. So, right. So we've got our donor cell with a plasmid in it that's got this cassette in it, yeah? And we've got our recipient cell that's got this Lac-I fusion. And what's going to happen is that when the plasmid gets into the recipient cell, it's carrying in lots of Lac operators, and the GFP Lac-I fusion will bind to the operators. Is that making sense? So look at this cell. Okay? So this cell has got the fusion diffused throughout it, but then what's happened is that suddenly you can see a spot appear, and the intensity in the rest of the cell goes down dramatically. So that's told you that this cell that started off without a plasmid in it has now got a plasmid in it. Okay? And if you go on watching that, what you see is that this spot replicates eventually, and you get two plasmids. Okay? So that is just a really neat way in which you can exploit these fusions. So I've almost got to the end, but I want... How many of you have heard of the yeast 2 hybrid system? Quite a lot of people, but not everybody. Okay. So the yeast 2 hybrid system is a way of detecting protein-protein interactions inside the cell. Okay? So the yeast galactose utilization system, in a way you can think about it a bit like the Lac operon, but it needs to be activated. And so we've got a promoter that needs RNA polymerase, but that's not sufficient. You need an activator protein that binds at an upstream activator site and consists of two domains. One is a DNA binding domain, and the other is an activator domain. So I've called them A and B. A is for activator, B is for binding. Okay? And the key thing is we've got our reporter here that tells us whether or not the promoter is on or off. And the simplest situation is where we actually take the open reading frames or the coding regions for these two domains and put them into plasmids, which allow you to make these proteins separately, these domains separately, and allow you to fuse those domains to other proteins. Okay? So imagine that you've got two vectors with those two domains, and you've fused them to the same protein, and that protein dimerizes, okay, interacts with itself. If it does that, then even though those two domains are being made separately, they can be brought together because the protein that they're fused to will effectively create a dimer. And that will bring domain A next to domain B, and you will get activation of this promoter, and your reporter gene will go on. Okay? And so the reporter gene is actually telling you, through the promoter it's been fused to, it's telling you about protein-protein interactions inside the cell. And you can obviously imagine the way in which, if you know that this protein normally interacts with something else and you don't know what that is, in the vector that makes encodes the activator, you can make a library of fragments. So instead of putting in the same gene, you can put in a library of loads of potential interactors, and then you can screen the library using this system for a second protein that will allow these domains to come together. Okay? And that is a similar sort of approach which is based on a different promoter. This is the PL promoter of lambda. You have a reporter. The key thing about this system is that it is normally very strongly repressed. If there is a lambda repressor there, the lambda repressor involves a DNA binding domain and a dimerization domain. You can make fusions between the DNA binding domain and other proteins, and that reporter will then tell you whether or not those other proteins dimerize. So if the other protein that you're investigating dimerizes, it'll allow this to become a dimer and still bind to its binding site and still repress the system. So your reporter gene, again, is telling you what is happening to this repressor protein. Okay? So there are lots of the same sort of principle emerging there. Okay, so that's the end of the lecture tutorial. Have you got any questions?
If you have a green fluorescent protein and you're measuring, say, like the light that's emitted, you said that it hangs around like for a long time. Like, how would you sort of trust, like, say, if you were doing, if you were measuring the light that was emitted, if it's hanging around? Like, how would you?
It's not, well, it's not that the light hangs around, it's the protein hangs around. And so it obviously depends on you what your worries are. I mean, as I said, you know, if you're interested in whether the system gets switched off, then and your cells are still growing, I mean, what you would expect in that situation would be that the amount of GFP per cell slowly gets diluted, but what you wouldn't see is the disappearance of the protein that had been made already. So that's where, so you should be able to then interpret the level of fluorescence versus the amount of growth there has been. That would tell you that there has been a switch off, but it would not be obvious until some time after, when you see this divergence between the levels. Does that sort of make sense? Yeah. In terms of the locational fusions, proteins should move around in the cell. They shouldn't, you know, the fact that it's not degrading should not mean that the protein will get stuck in a part of the cell and therefore cause that sort of artifact. So it really depends what sort of test you're trying to do. Any other questions? Well, if anything occurs to you during the morning, then please do ask, because this is, you know, this is supposed to be interactive. So, okay, I'm going to stop there. And look at what I'm supposed to. Yep.
When is that essay due in for this? Is it this year or is it in January?
I think it is after Christmas sometime. Yeah.
Is it when?
It is quite a long time from now. Yeah. Yeah.
We haven't had anything on this essay. Oh my God, what if it's due in a month?
Well, I think you have a choice of different, so it's Peter Wynne is in charge of this module, isn't he? So you need to choose, when you've done all these different methods sessions, you then choose one of the topics, and I think there is a fixed paper that you're supposed to review.